Good evening, everybody. My name is Sue Brewster, the Executive Director of Auckland Medical Research Foundation, now in its 64th year. And through your support, you have helped us to become the largest independent funder of medical research in New Zealand. You've helped to fund over $75 million worth of medical research projects, which is just amazing. And you've helped so many people. The fact that all of our administration costs are covered by a generous benefactor makes sure that 100% of your donation goes into medical research. And we're going to hear about some of that life-changing medical research here tonight. So Associate Professor Johanna Montgomery is combining her expertise in brain physiology with cardiac research, and Professor Julian Payton is looking at the control of blood pressure. Both Johanna and Julian have dedicated their working careers to this research, and the funding that's needed to continue this is all important. So we really can't thank you enough, our supporters who helped to make this possible, and your donation, no matter how big or small, is really vital. Your support really is so appreciated and very vital. Without further ado, I'd like to give a very warm welcome to Associate Professor Johanna Montgomery, who will present on making a difference in the world of abnormal heart rhythms.
Good evening, everyone. I chose this title, The Brains Behind Controlling Heart Rhythm, because it kind of also reflects my journey to this point. By training, I'm a physiologist, but specifically a neurophysiologist, interested in how neurons or brain cells communicate with each other. And of course, that happens within the brain, but the brain also talks a lot to the heart as well. So I'm going to tell you tonight about what's been going on in my lab and how brains actually help control our heart rhythm. But at the start, I'm going to tell you a little bit more about brain cells, because this is going to really help you understand it when I move to the heart. So here we have a really pretty picture of a neuron. So a neuron is a brain cell. Neurons are really unique kinds of cells. They're really different. You might remember from sort of high school biology, for a picture of a cell, you would have probably had a rectangle with a circle in the middle for the nucleus, and that was kind of the model of a cell. Well, neurons are really, really different cells. You can see from this picture, they have these really elaborate extensions that come from their central, what we call cell body. And these extensions are really, really important, because that means that neurons can form lots of networks between different brain regions within the brain. But also importantly, and more relevant for this talk, of course, is that neurons also extend these projections outside of the brain, all the way out to the periphery, including to the heart. So how do neurons communicate with each other? And they do that at structures called synapses. And so this little cartoon here shows us how synapses work. So here's one neuron or brain cell here, and it's sending out an extension here. And so this is the site of what we call outgoing information. So it's going to reach out and touch another neuron, and so this neuron here is the receiving neuron. And so if we zoom in on this area, this is a synapse, and this is how your brain cells are talking to each other. So one side that's sending the information will release bubbles of chemicals or neurotransmitters that's sort of speaking to the other neuron, and the neuron on the other side has these proteins called receptors here, and those are sort of like ears. They're listening to what that other neuron is saying to them. And so by doing that, neurons communicate throughout lots of different regions of the brain, as I said before, and we know that this communication is really important. This underpins how you learn and remember things, so this is happening a lot in your brain right now, how you move, how you sense. And one thing that's really, really a fundamental property of your brain cells is something called plasticity. So the synapses in your brain have plasticity. That means that that level of communication that your neurons are having with each other can actually change. And so that is what we call plasticity. So it's kind of like this fluid level that you can increase or decrease how neurons are speaking to each other. So for example, as I showed you before, we have this one neuron extending out here, and it may speak more to the other cell by releasing more of this chemical, for example, and or the receiving neuron might listen more. It might use its ears better, those receptors better. And so as a result, you can change the level of communication, and that happens, as I said before, when you learn and remember things in your brain. And we know that this process of plasticity in the brain goes wrong in a number of different disorders. So we know, for example, in Down syndrome, in Alzheimer's disease, Parkinson's disease, autism, all different brain disorders, that the actual plasticity that's happening at these synapses doesn't work properly. And what's been shown in the brain, at least, is that if you can basically what we call rescue that plasticity or make it return to what it normally is, you can actually reverse some of these clinical symptoms. And so that's kind of where brain research really is at the moment. And the reason we know so much about plasticity in the brain is that some really elegant techniques have been used to look at the neurons in the brain and look and listen to them communicating with each other. This is an example of the kind of work that we do in my lab, and this is something called electrophysiology. And basically what we do, so these are, right here, individual brain cells or neurons. And you can see these little glass electrodes coming down onto these cells. And what we literally do is bring down this glass electrode and suck it onto an individual cell. And if you do that to two cells at the same time, you can literally eavesdrop on what they're saying to each other. You can see the electrical signals that underpin that communication that these brain cells are having with each other. So that's an example of what's happening here on the right-hand side, where we see this spike here, so that's the cell that's releasing the chemical, and then these little downward deflections here, that's the other cell listening. And so this is their communication that's happening in real time. We can also do some really cool imaging in the brain, and it's got to the point now where we can actually do this in, for example, awake behaving animals. And so you can actually, so this is actually a microscope here that's mounted onto the head of an animal here, and what's happening is that the person in my lab is interacting with this mouse, and you can see each of these little dots here. That's a brain cell lighting up as a result of the communication that's happening. And so, for example, then you can look at different models of disease and understand how brain cells communicate with each other and how that's different in different diseases. But what about outside the brain? We know that there are lots and lots of neurons outside of the brain. In fact, some people say there are more of them outside the brain, especially in your gut, than there are actually inside your brain. We know that neurons project out into the periphery, including to the heart. And so this is the peripheral nervous system. And an example of parts of the peripheral nervous system are two systems, one called the parasympathetic and one called the sympathetic. Parasympathetic side of this peripheral nervous system is often referred to as the rest and digest part of your nervous system. So this kind of nervous system is really important for slowing your heartbeat, for example, for stimulating digestion, those kinds of things. Whereas your sympathetic nervous system, that does the opposite. That comes as that sort of fight or flight response with the release of noradrenaline or adrenaline. That results in your heart rate increasing, for example, inhibition of activity in the stomach because you don't want to be trying to digest food when you're trying to run away from a lion. So that's why we have these two balancing parts of what we call the autonomic nervous system. So if we were to zoom down of this peripheral nervous system into the actual heart, what we can actually see a lot of brain cells on the surface of your heart. Estimations of somewhere on the order of between 15,000 and 50,000 of them. And so this is just a diagram showing you from a review. Here we have the brain here, and we have a nerve here called the vagus nerve that's coming from the brain, and it comes down and basically forms synapses with each of these, see these little dots that are located on the surface of the heart. These are these things called ganglionated plexi, or little brains. And what they are basically clusters of brain cells all over the surface of the heart, largely on the atria. And so here, in this image here, what we've done is stained these neurons so you can see them. And so you can see it's really like a spider web or network of neurons that really coats the surface of the atria in your heart. So why should we care about these neurons? What do they do and how are they relevant to, for example, controlling heart rhythm? So one thing that we do know that's really important about these neurons is that they can regulate the rhythm of your heart. And clinically, it's been found that actually if you stimulate or activate these neurons, you can induce an arrhythmia called atrial fibrillation, or AF as it's often referred to. And to give you a little bit of background on what atrial fibrillation is, it's the most common serious abnormal heart rhythm. And currently, unfortunately, there is no cure for AF. Now, the actual rhythm that happens with AF in generally is not the dangerous part of it. It's the secondary effects that happen as a result of AF. And so what happens, so this is from the Heart Foundation of New Zealand. Normally, this is a sort of a normal electrical impulse that happens in your heart to make sure that the atria, the top chambers, and the ventricles, the bottom chambers of your heart, beat in appropriately synchronous manner. But in atrial fibrillation, what happens is you get a lot of these sort of random signals, a lot of excitation that's happening in places in the heart that it shouldn't be. And as a result, the atria contract very, very erratically and often very, very quickly. And what that does, what it can do is basically cause the blood to form clots. And so as a result, AF results in an increase in the risk of stroke by about 5-fold, an increase in the risk of heart failure by about 3-fold, and a doubling of the risk of dementia. And as I said before, this is the most common serious abnormal heart rhythm. Okay, so there are different forms of atrial fibrillation. Paroxysmal, which means that you have AF just intermittently, you're not permanently in AF, you just have it sometimes, and then your heart will go back to its normal rhythm. And then there are persistent forms when you actually have AF continuously. Some people are highly symptomatic of when they really know when they're in AF, they can really, really feel it. And for reasons we don't really understand, some people have no idea at all. And it can be picked up through a routine exam where a doctor may be listening to your chest for some reason or doing an ECG for another reason, and it will be observed that you actually have atrial fibrillation. So even though it's the same disorder, it can affect people really, really differently. And it can be, especially in symptomatic, or in both cases, can be extremely debilitating. So the treatment is very much sort of a standard approach. Patients will obviously often be put on a rate controller, such as a beta blocker, for example, a rhythm controller to help control the heart rhythm. There is a surgical procedure that you can get done called an ablation or a pulmonary vein isolation procedure, where they basically try and stop those impulses that I showed you before that are happening all over the atria. They try and stop those from happening by forming scar tissue to stop those inappropriate electrical signals from spreading where they are trying to go. And then usually patients will be on a blood thinner as well to decrease that risk of stroke. So there are things that can make AF more prevalent, risk factors as we say. So for example, how common AF is increases a lot with age. So it's highest in people aged over 75, so that affects about 12% of people, and these are numbers from New Zealand patients, as opposed to, it's patients in their 40s, which is a prevalence of about 1% of the population, 3% chance sort of in your 50s, 6% chance in your 60s. And there are things that can increase the odds of you having AF, as I said. So hypertension or high blood pressure that Julian's going to talk about next. It's observed in somewhere between 60 to 80% of hypertensive patients. Obesity also significantly increases your risk of AF as well. So coming back to these neurons of the heart that I said before, we think that they're really important in atrial fibrillation, but we really don't know a lot about these neurons at all, especially in humans. And I'll come back to this in a minute, but these are actually pictures of human tissue that we get from the heart surgeons over at Auckland City Hospital. And as you can see, so this is what these neurons look like in the humans. So you can see these dark red dots here. So each of these is that cell body of the neuron, the central part. And then surrounding that, this is all the fibres coming from those neurons. And you can see they're clustered together in this little, what we call ganglion. And here we've stained them with different dyes to show them up really nicely. So here you can really nicely see each of them in these green parts here and encased around in this ganglion here. And here are all those different fibres that are extending from and going to these neurons. So there's really a lot we don't know about these. And as I said before, I come from a neuroscience background. I'm really interested in how neurons talk to each other, how that communication happens, and then how that changes in disease. And we know from all that research in the brain that I briefly touched on at the start, that plasticity goes awry in a number of different brain diseases, like Alzheimer's or autism, for example, as I said before. So we're really interested to know, well, perhaps there's plasticity happening in these neurons of the heart, and perhaps they're changing how they're communicating with each other and how they're communicating with the heart. And maybe that is what is underpinning those electrical, those inappropriate electrical activity that's happening in the heart, and that may be what's causing atrial fibrillation. And that's the work that's going on in my lab at the moment. So we're wanting to know, does plasticity exist in these neurons? Can they change their communication? And why is this important? Why should we care about that? Well, as I said before, we know in the brain that if you can actually reverse plasticity that's gone wrong, you can actually change clinically what's happening to, usually it's in an animal model, but that's starting to be shown in humans as well. So we want to be able to see whether this can actually happen in the neurons of the heart. If they exhibit plasticity, can we then reverse that back and hopefully be able to restore normal functioning in the atria? So one way that we're doing that, and I'll talk about two different ways, one is in an animal model and one is in human tissue as well. So first of all, we do research in something called a spontaneously hypertensive rat. So these rats, this happens naturally in these animals, and they exhibit episodes of atrial fibrillation. So here's an example of the heart rate of one of these animals, and you can induce atrial fibrillation right here. So what we've done in my lab is basically take those technologies that I talked about at the start of the lecture that have been used a lot in the brain to basically decipher all the different ways that we know of that brain cells work, and we've taken that to the brain cells of the heart. And so this is an example here where you can see some of these neurons sitting here on the surface of the heart. And if you look closely, you can see one of those electrodes that I told you about before coming in here and sucking onto one of those brain cells on the surface of the heart. And so as a result, we can start looking at the activity of these neurons of the heart and trying to understand how they work and then how they may be contributing to atrial fibrillation. So one thing that we've found is that the neurons in the heart basically talk more in animals that have hypertension and atrial fibrillation. So what we can see here, so this suggests that they do actually have plasticity, because here in the red is the model that has hypertension and atrial fibrillation, and these black ones here are the controls. And what we can see here down below is we can look at the frequency of chatter. So each one of these lines that comes down is when the neurons are talking to it, either a neuron is talking to the cell that we're recording from. So we're basically, again, recording their conversations. And what we can see is that in the SHR, so the ones that are hypertensive and have atrial fibrillation, they talk much more than the other cells, the normal cells. The other thing that happens is that you grow more of these heart neurons when you have hypertension and atrial fibrillation. So here in the red are some pretty pictures. So if you look closely, you can see each of these red little dots there. Each of those is one of the brain cells on the surface of the heart. And what we find, if you look over here in part E, this is in the rest and digest flavour neurons. So the ones in that parasympathetic part of the nervous system that slows your heart rate. You see that there's an increase in the number of those neurons in animals that have hypertension and atrial fibrillation. But also equally, here in the blue, these are the ones that are involved in the sympathetic nervous system, that fight or flight response. What we find is that there's more of those as well. So hypertension and AF is changing the number of these brain cells on the surface of your heart. And this last little bit of data that I'm going to show from this work, if you look, each of these shows one of these brain cells on the surface of the heart, and you look at all these little dots. Each of those little dots is a synapse. So that's one site of where these neurons are communicating. And as you can see, there are lots and lots of dots in these pictures. So that gives you an idea of just how much communication is happening in these brain cells on the surface of your heart. And what we find is that the proteins that are involved in speaking and that's involved in releasing the transmitter are increased, and the proteins that are involved in listening, on the listening side of the synapse, are also increased with hypertension and atrial fibrillation. So far, our work is consistent with the fact that as well as plasticity happening in the brain, that it does also happen in the heart, in the neurons that are on the surface of the heart. And so what we find is that they are altered with hypertension and in animals that have atrial fibrillation. And so what we're trying to work on now is, as I talked about before, can we reverse these changes? And then if we can reverse them, will that then actually stop atrial fibrillation from happening? And that's the work that we're currently doing in the lab at the moment. And then importantly, how do we actually take this information that we're finding from an animal model and actually work in human cells? Animal models are a fantastic resource for research. But at the end of the day, we need to be able to cure these human disorders in human cells and in human models. But how do we do this? This is not as easy. It really isn't. So some of the challenges that face us when we're trying to look at do research in human cells is, of course, first of all, the low accessibility of human tissue. There are very strong ethical and cultural issues associated with collecting human tissue. And the other thing is, especially brain cells are really, really sensitive. They're really easy to kill, especially adult ones. And so you've got to treat them very, very specific ways to make sure that you can keep them alive. If we want to understand their physiology, we've got to keep them in top condition. And this is incredibly difficult for human cells. But having said that, we actually have been doing this in the lab for a while, and this is work in collaboration with Mike Dragonoff in the Department of Pharmacology, where for quite a while now, their lab has been collecting brain tissue from surgeries that are happening over at Auckland City Hospital. And we've joined with Mike and collaborating with him on the physiology side of this. And sort of to give you an example of the absolute importance of clinicians and scientists talking to each other, what used to happen, so for example, here is a patient who has a glioblastoma, unfortunately brain tumour in their brain, and you can see this tumour right here, and it's surrounded by normal brain tissue. And so what the neurosurgeons do is they go through this normal cortex to be able to to remove the tumour. And what they used to do was just take that bit of normal cortex and it would just get thrown away. Now though, that's not what happens. What happens is that they take the tumour out and we go over and we collect this piece of normal brain tissue. And then we can actually basically put the neurons in an enzyme solution to basically dissociate them from each other. And then here we can see how we can grow them in an incubator for weeks, even months. And these are beautiful, healthy neurons, and I can't emphasize enough how difficult this is. But what's really cool is then we can now start doing physiology on human neurons. So here's an example of using that technique that I talked about before, using that really fine glass electrode, and you can look at the communication that's happening between human brain cells that are growing. And you can see how chatty they are. Again, you can see a lot of these downward deflections, and you can see communication happening between these neurons. So we have been able to do this in the brain for quite a while. So actually, a cardiologist actually suggested, well, why don't you do this with heart samples? And we're like, well, okay, that would be fantastic. And so after about a year of paperwork, we got this going, and it's really, really exciting. So what we can do is get samples from these open heart surgeries that are happening over at Auckland City Hospital. So we go over, and here, this is the image that I showed you before. And so these neurons are embedded in the fat pads that sit on the surface of your heart. And so what the surgeons do is take a wee sample for us. And this is an example of, as I showed you before, this is the neurons here and surrounded by the fat that we've managed to stain as well here. And then here you can see this is basically showing up all the synapses, so all the sites of communication that are happening between these neurons. So you can see in human cells, there is a lot of communication happening between these neurons as well. And so what we want to do is, yes, other people have been able to take pictures of these cells, and that's fantastic, but we want to actually know about their physiology. We don't know anything, literally, about the physiology of these neurons from the human heart, because no one has ever recorded from them before. And so what we want to be able to do is actually study these neurons, do some physiology on them, and really try and decipher what their properties are, and actually have a look and see how they might be different in patients, for example, that have atrial fibrillation versus not. Because that can give us really important clues as to what may be actually underlying that abnormal electrical activity that's happening in the heart. And so if we can manage to do that, and I can tell you that we have managed to do that, but not as often as we would like to just yet, but every day we're making progress on this. But this was from our very first recording, successful recording, I should say, that we've done from these neurons. So it is actually possible, incredibly difficult, but possible, to do electrophysiology on these human neurons that we get from human heart patients. And so we're really excited about what's going to happen over the next year or so, and hopefully we can update you on what might be happening and whether we can really see some differences in the properties of these neurons with these disorders. Now, just stepping back a bit and sort of going a bit more international, neurons are really becoming a major focus of developing a lot of different treatments for atrial fibrillation. I'm going to give you two examples of that happening now. So one of these is a neurostimulation therapy, and it's a trial that's happening, and it's called the TREAT AF trial. And so what is involved here is literally putting a little stimulating device on the tragus, this little lump here on the side of your ear. And what happens there is that electrical stimulation was given to patients. They sat for an hour every night for 6 months, so this took quite a bit of commitment. But what that does is it stimulates that vagus nerve that I talked about before, that one that goes from your brain down to your heart. And there's information that goes in both directions. And what they found in this was a very small trial. They're about to expand this out a lot more. But they found at 6 months that the burden of AF was reduced by 85%. So what they found is that it worked really, really well in some people, but for some reason that they don't understand, it didn't work at all in others. And they're trying to understand why that may be. But Stavros, the lead scientist and clinician, basically this is a quote from him saying that he thinks that their clinical trial introduces a novel, self-administered, non-invasive therapy for patients newly diagnosed with atrial fibrillation. And what is really exciting is that actually we're going to be getting some of this tissue from patients that have had some of this stimulation. They're going to be sending it from the States to us so we can do the kind of images and physiology that I talked to you about earlier, like we do for the patient samples from Auckland City Hospital. Another thing that's sort of a hot new area, and believe it or not, it's getting Botox but in your heart. And so what actually happens is that what they've done is inject Botox into these fat pads where these neurons are localised on the surface of your heart. And what they've been doing is doing this on patients that have bypass surgeries. And the reason they've been doing that is because there's something called post-operative atrial fibrillation. And that's very common in people who have had bypass surgery. And they want to be able to obviously decrease the burden of that because then that will significantly decrease the risk of stroke after these kinds of surgeries. So what they've done is basically inject the Botox in. So what does Botox do? It actually stops, how I talked about how neurons communicate, and the one that's speaking releases those little bubbles of chemical or neurotransmitter. So what Botox does is it actually stops that. So it stops the neurons from actually being able to chatter on that side of the synapse. And so it stops them speaking. And what they've found, and now there's actually just been a paper that's come out very recently showing that they've followed these patients now for 3 years post-operatively, and they've found a sustained and substantial decrease in AF during that whole 3 years. So it's going longer and longer from this initial, and this was just from something that they did right after surgery. So these are some really promising therapies that are happening. So neurons really are becoming a major focus of treatment for AF. And lastly, I want to talk to you about another way that we can study human cells, and that's looking at another different kind of heart arrhythmia, an abnormal heart rhythm called long QT syndrome. And what happens with long QT syndrome is that the heart takes longer than it should to be able to recharge or relax during beats. And so what happens is that another electrical signal will come along, but the heart hasn't relaxed properly yet. And so as a result, it can result in a really rapid, dangerous heart rhythm. And unfortunately, this is the leading cause of sudden death in the young. And so Annika Wimbo, a research fellow in my lab, is leading this work. And what's actually happening here is we're using blood samples from patients who have long QT syndrome. And what is really, really cool, and this is work that was done obviously outside the lab, was Nobel Prize winning work showing that you can actually get cells from the body and turn them back into stem cells. So you can take, in this example, we take blood cells, you can turn them back into stem cells. And then once you've turned them back into stem cells, you can then differentiate them into different kinds of cells in the body that you're interested in studying. So for the purpose of this study, we can turn them into heart muscle cells, or we can turn them into sympathetic neurons, so the fight or flight flavour of neuron that innervates your heart. And we can do physiology, and these are the patient's own cells. And what's fantastic about that is then you can feed back that information back to the patient, and ultimately we want to be able to influence their treatment because we have information specifically on how their own cells are behaving. And so right here, this is a very cool movie, and if you'd asked me this 5 years ago, would I ever have beating heart cells in my lab, I would have said, I thought you were crazy, but it's so cool that these were blood cells, not long ago, and now they're beating heart cells. And what we can do, so this is what the heart cells look like when you're looking at them in a still image. And then these are the sympathetic, or those fight or flight heart neurons. They have a really distinctive sort of shape, very round cell bodies, and very thin, long processes that extend from them. So these have all been made from a patient's blood cells, and now they are heart cells or they are heart neurons. And so what we can actually do is grow these together. And this is really important, is that from now, we've really got to start integrating different systems in the body. The heart alone, so it can't be treated alone. Lots of things influence your heart and vice versa. And it's really important that we start looking at how other factors influence what's happening, for example, in the heart. So we can actually grow these human heart cells, heart muscle cells, and heart neurons together in a dish, and we can look and see how they're communicating with each other and how that might be different in a patient with long QT syndrome. What are the neurons doing that's different as opposed to a person who doesn't have long QT syndrome? And then we can actually look at different strategies for treatment using those patients' own cell. What works for this patient who has this specific genetic variant versus another patient who has a different kind of genetic variant? And so this is really sort of one example that's bringing us towards an era of personalised medicine, which I think we're going to hear more and more about as research goes forward. No one is identical. One treatment will not be the same for everyone. It will affect all of us differently because we're all genetically different. And it's really important to realise that and to start incorporating that into our research. And I'm just going to finish on a personal note. This is something that's affected me, and I want to talk about that a bit more. This is my dad, and he was meant to be here tonight. And when I was invited to give this talk, he was really excited, and he wasn't a scientist, but he just loves learning about science, and he was affected by hypertension that was very hard to control, and atrial fibrillation as well. He was really excited to come here tonight, so unfortunately, about 6 weeks ago, his body had other ideas. And I share this because, first of all, I wanted to dedicate this lecture to him, and I didn't do that at the start of the lecture for obvious reasons, but also because I wanted to share the fact that I think pretty much everyone in this room can have a similar story. I think it'd be really hard-pressed to find someone in this room who hasn't directly or indirectly been affected by heart disease. And what's fantastic is research has moved us a long way, but we still have a long way to go. And I just want to thank you all very much for contributing and helping with this. And hopefully we can make more differences for more families in the future. Lastly, I just want to thank this fantastic group of people that I get to work with every day. They really are the loveliest people. And specifically, I want to thank Jesse, who's played a major role in getting this work started in my lab. As I said, my background is in neuroscience. And he really helped bring the heart work, just he's up the back there. So really helpful and really grateful for him for getting this started in my lab. And he's now been joined by Liam and Jocelyn, who are helping him in doing that. Annika's leading the work in the stem cell research. She's also up the back there in my lab with the help of Sugi. I want to thank my collaborators a lot, especially the patients over at Auckland City Hospital who are enabling us to do some of this human research. That is really lovely. All the clinicians, they have just been fantastic at helping us recruit the patients, give us the samples. And also really a huge, huge thank you to all the funders, both past and present, who have enabled this research to start happening in my lab, and especially the present funding from the Marsden Fund, from the Auckland Medical Research Foundation and the Freemasons Foundation. That's enabling us to take this work to the next level and hopefully be able to really uncover what's happening in terms of plasticity in neurons and atrial fibrillation. And I'll finish there, so thank you so much.
I'd now like to introduce our second presenter for today, and that's Professor Julia Payton, who is looking for answers in controlling blood pressure and preventing life-threatening medical events. Welcome, Julia.
Tēnā koutou, I tēnī ahi ahi. Ladies and gentlemen, welcome to my lecture this evening, where I'm going to talk about blood pressure and some novel ideas about controlling it. Thank you very much to the Auckland Medical Research Foundation for this fabulous opportunity. One of the reasons that it's fabulous is because the first time now, my dear family sat here. I've got their entire concentration for 30 minutes. They're now going to find out, without interruption of such devices, what Dad actually does all day long. Blood pressure, how we might fix it. Let's start with some facts, which are really rather depressing, but I can assure you it'll warm up afterwards. It's the single largest cause of mortality worldwide. 750,000 of us in New Zealand suffer from high blood pressure. This room holds around 250, 260 people. There are at least 60 to 70 of us with high blood pressure in this room, according to that statistic. Hypertension-related mortality is disproportionately higher in Maori and Pacific people, and that's something that needs urgently addressing. It's known as the silent killer because you can't feel blood pressure, and that's why it's so important to get it checked. In 95% of the cases, if you ask a doctor the cause of somebody's blood pressure, they will tell you, I don't know, it's unknown, it's idiopathic. Remarkable. 50% of the strokes and heart attacks could be prevented if only we could control blood pressure. Why is it so tough to control? Why should we care about blood pressure if it doesn't hurt us? It's not like arthritis, that every morning we get a tweak or a pull or a pain that reminds us to take our pills. Blood pressure doesn't remind you to take your pills, and so people often forget to take their pills. The pills cause symptoms, and if you start developing symptoms from an asymptomatic condition, why take them? But it drives stroke, it drives heart failure, and it drives heart attacks. It drives kidney failure, it drives vascular dementia, and as we've heard, it causes atrial fibrillation. All of these potentially are killers. We need a massive team effort to fully understand the causes of blood pressure, which are, of course, multiple reasons. Such a team may be the All Blacks, or maybe the All Whites. Now, I realize I'm on thin ice, and I'm going to quickly move away from rugby, back to blood pressure, and define blood pressure as simply the force exerted on the arterial walls in the body. Too much force is a bad thing. The first measurement of blood pressure was 1733. I think one of my most heroic studies of all time. 1733. Go back to 1733. That's a long, long time ago. Measurements were made in conscious horses. How do you measure the blood pressure from a conscious horse? There is no anesthesia, there's no sedation. Well, it's obvious, right? You use rope and a wooden gate. You lash the animal down, and then you have to think, what tube do I insert into the carotid artery to measure blood pressure? Pressure enough to defy gravity and raise blood, which after all is quite heavy, above the ground to a distance, as you can see measured by the Reverend Stephen Hale's choir boy here. The tube should not kink. So what did he use? A windpipe from geese, stitched together by his dear wife and inserted into the common carotid artery. Pressure rose to a height that defied gravity. And the only reason that it wouldn't go higher was because of the opposing effect of gravity on the mass of blood being pumped. So if you wanted to generate higher pressure, you would have to increase the force of the pump. Gravity and blood pressure is something I will come back to. Now, blood pressure is pulsatile. And it's pulsatile because the pump that generates pressure squeezes, as you know, as you've heard from Johanna. And there is a systolic blood pressure, which is the high one, 120, and the other number is the diastolic blood pressure, which is the time when the heart is relaxing and filling. And it's that squeeze that generates that upstroke of pressure to give you the pulsatility. And here is a pressure manometer, and it just so happens we have a pressure manometer that is identical. And it just so happens we have a subject. And it just so happens that I'm crazy enough, under conditions of high stress, it has to be said, looking at all you people, that's going to cause fight or flight types of reaction to record my blood pressure in front of you. First time I've ever done this. We'll see what happens. Now, the blood pressure on this graph shows 134 over 84, and I've chosen that because on Sunday night, as I was thinking about what I would say, that was my blood pressure, and I was quite happy with that. Right now, we're going to see what my blood pressure is, and it tells me it's got an error, and that's probably because I'm talking too much. So I may have to shut up in a minute. But if my blood pressure exceeds 140 over 90, then I am hypertensive by definition. That is accepted by the world. Let's go back to the cuff. What is it doing? Because it's quite informative. What the cuff is going to do is it's going to squeeze my artery. I'll keep my arm still this time. It's going to squeeze my artery, and it's going to stop the blood from flowing into my arm. In the cuff is a sensor for oscillations. It detects vibrations caused by the pulsatile nature of the blood flow. Those oscillations disappear when you exceed systolic pressure. The cuff pressure then releases slowly. I'm sure you've had your blood pressure taken. And once the oscillations have disappeared, that is because you've completely opened the artery and flow is laminar, and you don't get those oscillations. And that's how this device works. It can also tell you your heart rate. We'll just wait until it's decided what my blood pressure is. The reason it's taking a long time is because it's very high. My goodness, you don't want to know what that is. Moving swiftly along. Blood pressure moves, and it moves dependent upon state. So depending on your behavior, your blood pressure will change. And that is perfectly physiological, because there are conditions where your blood pressure needs to change in order to direct different amounts of blood flow. But it always comes back to a resting point. Now, the healthy person, indicated here, has what we call ambulatory blood pressure. It's a device like that, but it's pumping up every 30 minutes and taking a measurement every 30 minutes for 24 hours. And unfortunately, it keeps you awake at night. But you can see that during the day, it is bouncing around all over the place. But it always comes back to where it should be, at around 100 millimeters of mercury. A hypertensive patient has a consistently elevated blood pressure. As you can see, again, it can undergo excursions. And there is no dip, as you see here, which is a sign of good health in blood pressure at night. Now, a man that's under pressure at the moment is Boris. And let's assume he has a relatively normal pressure of 120 millimeters of mercury. But the point of this slide is, what is 120 millimeters of mercury, really? And that's why he's not a scientist. But 120 millimeters of mercury is equivalent to 2.4 psi. You have about 28 psi in your car tire, and you require in the region of 75 millimeters of mercury to blow up a balloon. That gives you some idea of the sorts of pressures that we're dealing with here. Why do we need blood pressure? We need blood pressure to generate blood flow. And blood flow is by far the most important physiological variable. The reason we measure blood pressure is because it's relatively simple. But it's the blood flow that the organs care about, because it's the oxygen and the nutrients that they require. And in order to get flow, you need a pressure head. Probably the most precious organ is the brain, and it requires lots of blood flow because it has the highest oxygen demand, because it's so metabolically active. And so subserving blood flow to the brain must take priority. Now, normally blood pressure rises with age, as you can see here. This is perfectly normal, and I'm in this bracket here. Okay? And as we age, our cardiovascular risk increases, which means we're more susceptible to heart attacks and strokes. But the cardiovascular risk will increase even more if we also smoke, if we also drink, we're overweight, we don't exercise, we have high cholesterol, we have high salt, and we have a family history. These are all additional factors that potentiate the cardiovascular risk from happening. Don't you love that slide? I mean, we know exercise is good. Why would you have an escalator going into a fitness center is beyond me. If you go to your GP and he or she tells you you have high blood pressure, the first thing they'll say is change your lifestyle. And can you imagine a life without all of that? It's really difficult to change your lifestyle, but to change it where you then have to start meditating, you cut back on milk chocolate. I couldn't understand a life without milk chocolate, quite frankly. Reducing alcohol and salt, and so on and so forth. But imagine if we could study a population of people that were really prime specimens, that didn't have all those issues. And I'm going to take you to the Amazon. I'm going to take you now to Venezuela and Brazil, where there are two tribes, the Yanomami tribe and the Iguana Tribe. They are completely isolated from the world. They live in a beautiful natural environment. Let's have a look at their blood pressure. The Iguana Tribe have blood pressure that rises with age, as I just showed you. But the Yanomami tribe do not have blood pressure that rises with age. And why is that different? What can we learn? The Yanomami tribe that do not have the rise in blood pressure with age have a wonderful diet. They have a 20th of the amount of salt we ingest. They eat vegetables and fruit. They exercise. They don't smoke. They don't drink. They drink water from a stream. It's almost unheard of. It's unreal. But what happens with the Iguana tribe, who are just next door? They have the same environment. Everything is the same, except one thing. An airstrip. Invasion of Western influence. Smoking, alcohol, high cholesterol, high salt has been brought into that tribe, and as a result, their blood pressure goes up with age. Now, what do fridges, kidneys, and Akasiga all have in common? Akasiga is the first vertebrate onto land. It's salt. Refrigeration was able to remove the need to use salt for preserving food. So salt ingestion went down. Of course, the kidneys removed salt. And this critter here invented ways in which it could preserve water and salt within its body. If it's leaving a wet environment, it's got real problems of facing the consequences of dehydration. And it invented something, or evolved something called angiotensin, which is a hormone. And angiotensin is one of the biggest culprits of cardiovascular disease. And if you don't believe me, then if you look at the ABCD of treating blood pressure, it's all around predominantly treatment of angiotensin receptors, as we heard about receptors earlier. It's all about salt. So we have angiotensin blockers that increase, that prevent or cause more excretion of salt. We have beta blockers that reduce the amounts of angiotensin. And we have diuretics which help remove water and salt. And there are other things, calcium channel blockers, that aren't involved with controlling salt. So clearly salt's important. But look at this statistic. 50% of patients that have high blood pressure that are on treatment remain hypertensive. What does that mean? Hypertension is more than just salt. Some people are salt sensitive, others are not. Again, we come back to this issue that Johanna mentioned. One treatment does not fit all. We have old and young people that are hypertensive. We have black people and white people. We have men and we have women, different ethnicities. We cannot use the same medications to treat these different populations. Yet over the world, what do? We follow A, B, C, D. And if one of those monotherapies doesn't work, you add another, and then another, and then another. And is it any surprise that we get side effects and don't take our pills? I don't think we are treating the cause. I think in most part we're treating the symptoms. We have to get to the root of the problem. So the question I pose is, what else controls blood pressure? The brain. We're back to the brain, the master controller, the orchestra of the concert. The brain is connected to the heart, as we've heard, and it's also connected to every vein and artery in your body. And we're now going to demonstrate, with the help of a young individual who I've never met before. We're going to demonstrate the connection of the nerves between the brain and Sebastian's heart. Okay? And you heard earlier about the sympathetic system, the fight or flight system, that increases your heart rate. And you heard about the parasympathetic, the rest and digest, which decreases your heart rate. The brain is intimately controlling heart rate, and that beep is Sebastian's heart rate. And what I want you to listen for is the change in the frequency of heart rate as he breathes. Because every time you inflate your lungs, still alive? Every time you inflate your lungs, you change your heart rate. So it'll go up during inspiration and down during expiration. He's going to raise his hand up when he breathes in and down when he breathes out. And hopefully you will hear a change in the beep. He has very profound respiratory sinus arrhythmia, a naturally occurring arrhythmia. It's a sign of good health. It's classically seen in the young and the fit, and Dad doesn't have it. So arteries and veins are covered in nerves. These nerves cause constriction. The constriction that you can see operating here is a result of their activity. The more activity in these sympathetic nerves to the vessels, the higher your pressure goes. And we believe that sympathetic activity is a target for controlling blood pressure. The reason is that all the current frontline medications, the ABCD, do not affect sympathetic activity. This is a beautiful image of those sympathetic nerves, and you can see the density around a blood vessel of those particular nerves. And just a small change in the diameter of a vessel can have a dramatic effect on the resistance. Now, Julia Roberts happens to be my favorite actress. If we look at somebody that's standing upright, their blood pressure is affected by gravity. I mentioned that before. Classically, you're around 100 millimeters of mean pressure. It's slightly less at the base of the brain, and it's higher in your feet because of gravity. Quite a dramatic effect. So if gravity is affecting blood pressure, think of the giraffe. It is the most unbelievable physiological animal because it is pushing physiology to the limits. This long neck poses a serious problem of how it generates blood pressure to its brain, the most vital organ. If we resize Julia Roberts to the same scale as the giraffe, and we calculate how high her heart can pump blood up that neck, you can see it barely gets to with 1/5 the way up the neck. How has the giraffe then, which has been on the planet for 2 million years, evolved to increase its pressure? Can we learn something from this? Well, its pressure is high. It's physiologically hypertensive. It doesn't die from cardiovascular disease. And it's that pressure that allows it to push blood up into its brain. And also note, remarkably, that the level of pressure at the neck, at the base of the brain, is identical to yours and mine. That has been preserved. Why is that? Because it's the optimal pressure for oxygenation of the brain. Now, if we look at the foal, when newly born, has the same pressure as you and I, but the pressure elevates as the neck elongates in the giraffe. And this led to the idea that there must be a sensor high up in the neck that detects blood oxygen, that generates high pressure in the giraffe. Could that sensor exist, and could it cause blood pressure in humans? Well, such an organ sensor does exist. It's called the carotid body, something that's been keeping me occupied for 35 years. It's the size of a rice grain, and it looks like my mother's red currants. It is a major oxygen sensor. It's a guardian of blood flow to the brain. Its nerves connect directly to the brain. Yes, when activated, it can increase pressure profoundly, and it also drives the sympathetic nervous system to raise blood pressure. It vasoconstricts and causes increases in heart rate. It has the body's most profuse blood supply, and I'm just showing you here what looks to be a pile of spaghetti. But this is, in fact, the vasculature, the vessels within that little structure, that little organ. It is very capable, therefore, of tasting, sensing the blood with that enormous blood supply. So we asked the question, if we took an animal with high blood pressure and disconnected the carotid body from the brain, would blood pressure change? And yes, it did. We got a whopping fall in blood pressure when we removed the carotid body's input to the brain. That was the first realization that the carotid body could be a new target to control blood pressure. In other words, these small organs are screaming to the brain to elevate blood pressure. What would happen if we could remove the carotid body from a human that was resistant to drug therapy? Could we reduce their blood pressure? So we studied this in 16 patients that were on at least 6 antihypertensive medications and still had systolic pressures of over 180 millimeters of mercury, truly resistant. This is the first carotid body removed from one of those patients, and you can see it does look like my mother's red currants. I wasn't lying. And look what happened to blood pressure. We got a whopping fall in ambulatory 24-hour blood pressure of 22 millimeters of mercury that was sustained for 6 months. And that fall in blood pressure in many of the patients meant that those patients had to come off their antihypertensive medications because blood pressure was falling too low, too quickly. 60% of patients studied responded. This was the first in-human safety feasibility study in the world. But it's no good advocating cutting out carotid bodies because carotid bodies are very important and they do lots of other functions which we do not want to lose. So it was up to us to work out why it is the carotid body screams. And we discovered that it releases a molecule called ATP. That is the energy molecule that we all rely on for driving energy, contracting muscles, etc. But it's also a transmitter, very similar to what Johanna was talking about, a neurotransmitter that acts on receptors called P2X3 receptors. And it's through the activation of those receptors that drives information to the brain that increases blood pressure. And these P2X3 receptors are indeed found in human carotid body. These were extracted from cadavers. And the P2X3 receptors are labeled in green. They are everywhere in cadavers that had a history of hypertension. So we knew that the target is present in humans. This got the attention of a small pharmaceutical company that had a highly selective P2X3 receptor blocker called gefapixant, GPT. We gave gefapixant to our hypertensive rats and we got a 28 millimeter of mercury fall in blood pressure, which was fantastic news. Now not only we had found an organ, but we'd now found a way in which we could block the transmission, the signaling, the screaming to the brain to lower blood pressure. On the eve, and this is not a word of a lie, on the eve at which we were going to submit our ethics application to give gefapixant to human hypertensives, Afferent Pharmaceuticals pulled the plug on us and told us that they'd sold out to Merck for multi-million pounds. The trial was all over. Merck had no interest in blood pressure. This has been a roller coaster. This is a real roller coaster. This is really a great shame. But the good news is that after 4 years, I have persuaded Merck through some negotiation to test the silver bullet therapy, as I call it, for treatment of blood pressure. And they finally set up a trial. They're mainly interested in sleep apnea, but many of the sleep apnea patients will have hypertension. We should hear more in 2020. I want to focus on another area that is gaining huge traction as a new way to control blood pressure, and that is through the stomach, the gut. We know that in the human gut, there is somewhere in the region of 5 to 1,000 species of bacteria. And there are 100 trillion bacteria cells in our gut, and that's 10 times more than there are body cells. Remarkable, absolutely phenomenal. And these bacteria live very, very happily. They're friendly bacteria. They cause no immune response because there is a functional barrier between the gut and the blood. So there is no immune response. But in diseases such as hypertension and heart failure, and many others, we now know that this barrier breaks down and that these cells, these bacterial cells, cross into the bloodstream and produce an immune response. And that immune response, apparently, can cause disease, such as high blood pressure. Now, this is a very recent study taking the feces from a normal rat with normal blood pressure and giving it to a high blood pressure rat. And when I first saw this, I thought, oh shit. But actually, they don't eat each other's feces, but actually rats do eat each other's feces. Dogs also eat feces, and you've always pulled them away, but maybe you shouldn't. Because contained within feces, there are bacteria. Here are a couple that we now know are probiotic. These are the good ones. These are the ones you want, even the smelly ones. These ones actually exist in your dog's mouth. So if you kiss your dog, that is probiotic. If you if you feed your gut with the right bacteria, look what happened to the rats. You can lower their blood pressure. That is a new way of lowering blood pressure. Feed yourself with the right gut bacteria. If you choose the wrong bacteria, and if you have normal blood pressure and you eat bacteria from feces from a high blood pressure rat, you will generate hypertension. It works both ways. Now, turns out that the gut bacteria get into the bloodstream and they affect the brain. And it's through, again, activation of the sympathetic nervous system that puts blood pressure up in those animals. And we started a study some time ago using immunomodulatory therapy. And immunomodulatory therapy could also be on the cards for treatment of high blood pressure. We use something called minocycline, which is a broad-spectrum antibiotic. But the special thing about minocycline is that it crosses into the brain, and it can get into the brain and reduce the inflammatory processes that are occurring in your brain. And by treating patients for 1 month on minocycline, we got whopping falls in blood pressure and reductions in sympathetic activity. I do feel that immunomodulatory therapy will come about for certain individuals with high blood pressure. But it's all about proper diagnosis. So I'm going to conclude, and I'm going to tell you that we have to have a fully integrated response if we're going to understand blood pressure. You simply can't rely on this alone. You have to involve the carotid body. Yes, I'm biased, but I've spent a long time looking at it. You have to involve the immune system. You have to involve the kidney and the salt and the heart and the microbiome. And the high blood pressure will vary between individuals. It's a consequence of multiple mechanisms. These mechanisms are not understood. And that diagnosis and treatment should be more integrative, and as Johanna concluded, should be more personalized. I need to acknowledge an awful lot of people, some of whom are here. I apologize for those that I've not included, but the home team over here, my new home team in Auckland, have been absolutely wonderful in receiving this crazy Brit to come and set up operations, and I'm very grateful to the really warm reception that I've had. My international collaborations, many of whom are back in Bristol still. I'm still working with many of them. I want to thank many of the funders that have recently funded some new projects for us, as well as the British Heart Foundation, who funded me for over 30 years. And I want to thank, again, the Auckland Medical Research Foundation very much. I want to thank the University of Auckland for accommodating me, and I especially want to thank you all very much for listening to me. Thank you.